This is Mr. Jones, and the purpose of this video is to explain how to do the DNA scissors activity for biology. And so the DNA scissors activity is helping us learn about what are called restriction enzymes. Restriction enzymes are bacterial enzymes whose job it is to cut up DNA, and their real job is to keep foreign DNA out of bacteria. But they can also be used in biotechnology to help us insert genes, say, from one organism into a different organism. And so the way that restriction enzymes cut up DNA is not just randomly chopping it up like, say, what might happen in your stomach or intestines when you're digesting food, but rather it is cutting the DNA at very specific locations based on the DNA sequence. And these are what are known as palindromes. So let me scroll down here and we'll talk about an example of this. This is the recognition site for what's called the EcoR1 enzyme, restriction enzyme, and it is looking for this specific sequence, GAATTC. And that's called a palindrome because a palindrome is something that is the same in forward as it is in reverse. And when you look at the complementary strand for the GAATTC, what you notice is that if you read it again from 5' to 3', it also reads GAATTC. Now, what happens when the EcoR1 enzyme finds this sequence? It cuts between the G and the A on this upper strand, and it also cuts between the G and the A on the lower strand, and then it breaks the hydrogen bonds between the ATAT, TATA, and this is sort of demonstrated down here. And what you wind up with when you're done is two pieces of cut DNA. Now there's much more sequence way over here, and there's much more sequence way over here, but these are what are known as sticky ends because once it's cut, one of the strands of DNA has 4 bases overhanging the end of the other strand of DNA. And this will become more clear when I demonstrate to you how to do this actually using scissors on some specific sequences. Similarly, over here, you've got this upper strand with 4 bases overhanging where it was cut on the lower strand. So that is an example of what is called a sticky end. And these are called sticky ends because you have these extra bases overhanging that could actually have complementary bases come in and stick to it using hydrogen bonding. There's a different type of cut that's called a blunt end cut. And so an example of that would be SMA1, which is looking for this sequence, CCC-GGG. And when that happens, it actually cuts between the C and the G on each strand, which are directly opposite each other. And so it cuts all the way through, and you don't have the overhanging. You have what's called a blunt end. So let's take a look at the assignment and the sequences that are on the 3rd page, and let's take a look at what it's asking you to do. The first thing it's asking you to do is we scroll down to these exercises and questions. It's asking you to take strip number 1. So now I've got a PowerPoint presentation that I put these pictures into, and I'm not going to show it in slideshow mode. This is strip number 1, and the first thing that it asks you to do is to cut that strip number 1 with EcoR1. Well, remember, EcoR1 is looking for a sequence that is GAATTC. And so that sequence is right here in strip 1, GAATTC. And you notice on the bottom, it is also GAATTC. So I've just kind of marked that with a pencil. In each case, it is going to cut between the G and the A. It's going to cut between the G and the A on the top strand, and it's going to cut between the G and the A on the bottom strand. But it's not going to cut all the way through to the other side. It's just going to cut there, and then it's going to peel back these hydrogen bonds. So when you actually make that cut between the G and the A on the top and the G and the A on the bottom, and then cut through the hydrogen bonds, you get 2 fragments that look like this. Once again, these have what are called sticky ends. You'll notice that there's this TTAA overhanging on the bottom, and there's this TTAA overhanging on the top. Now, when you go on to point 3 under exercise 1, it tells you to do the same thing now with strip number 2, except now you're going to cut this with SMA1. And if you'll recall, SMA1 is looking for the sequence CCCGGG. So we have found that CCCGGG. And now I've marked where it's going to cut. It's going to cut between the C and the G on both points. When I cut, now I cut all the way through and I'm left with what are called blunt ends. Okay, now I've done the first two. Now number 4 through 5, points 4 and 5 are asking you to do this again on strips 3 and 4. It asks you to use an enzyme called Hindi3 on strip 3, and then to use EcoR1 again, which we used on strip 1, on also strip 4. Now here's the thing about sticky ends when they occur. They can be stuck back together. So here's the two pieces. So when I cut strip 1, it looked like that when I was through cutting. But I can actually put them back together, and you'll see that everything matches back up complementarily, A with a T, A with a T, T with an A, T with an A. And there's an enzyme called DNA ligase, which will actually reform these GA bonds and put everything back together. And so points 6 through 9 are asking you to look at some of the cuts you've made to strips 1, 2, 3, and 4, and see which pieces from which strips might be able to be reconnected to other pieces from other strips, which is simulating the way actually that we might be able to insert, say, a gene from one organism into a different organism using these restriction enzymes and DNA ligase. Now let's look at the second part, exercise 2, which is asking you some questions about a plasmid. A plasmid is a piece of bacterial DNA that is actually in the form of a ring. It doesn't have two ends. It's just a continuous ring of double-stranded DNA. This particular one is called YIP5, and it is 5,541 base pairs long. And so this point on the plasmid indicates where we start counting 1, 2, 3, 4, 5, 6, 7, 8. And we would count all the way around the ring until we got to number base 5541, in which then we would be back at base 1 again. Remember, like all DNA, this is double-stranded. So it's a double helix going all the way around this ring. And so the first question, question 10, asks you what would happen if I digested this. That is, if I cut it using enzymes EcoR1 and EAG1. Well, it turns out that they have marked for you where the specific restriction enzyme sites are in this plasmid. And so if I were to digest this, that is, cut it with EcoR1, it would cut between base 5,541 and base 1 right there. So if all I did was cut this with EcoR1, I would simply go from having a ring, the DNA in a ring, to having a situation in which I have a 5,541 base pair long DNA sequence. But now it has been cut there, so it's just like a chain that has been opened. But if I cut this with both EcoR1 and EAG1, which is what number 10 asks you to do, I'm actually going to wind up with two fragments, right? Because I will have cut it here and I will have cut it here. And now to say how long are those fragments going to be, we're just going to do math. The first fragment is going to be from base 1 to base 942, and so that fragment is going to be 942 base pairs long. The other fragment, I can simply do the math and take 5,541 and subtract 942. That gives me 4,599. So I will have one fragment that is from here to here. That's 942 base pairs long. The other fragment is going to be the rest of it, going from this point all the way around the ring back to the starting point. And that fragment is going to be 4,599 base pairs long. So that's what happens for number 10. Number 11 asks you to digest it with a couple of different enzymes. Hindi 3, which starts here at base 32, and EAG 1, I'm sorry, with APA 1, which is right here, APA 1. So that means you're going to cut a fragment from here to here, and then your other fragment's going to be from there all the way around and back to here. And again, you can work that out using math. And then number 12 asks you to actually cut it with 3 different restriction enzymes, which would give you 3 pieces of varying lengths. And so you can probably work that out for yourself and do the math to work out what the length of the various pieces would be that you would have once you've made those digestions. Okay, well, that's a description of how to do the assignment, and I hope you found it useful.